Good afternoon, everybody. Hope all is well. Hope you had a great weekend. Certainly was a pretty weekend. All right. If you go to Canvas, I go to discussions. I downloaded a worksheet for you. I'm going to go over it with you. There's 99 questions, but I sent you the answers. And so this is a review page that I got from quizzes that should help kind of generalize everything on one page. And again, all we can do is just give you practice questions. We don't know what's actually on the test. But if you go to discussions, it's under the discussions. It should come up like that. I'm going to stay in and just take a look at it while we wait for others to come in. I will go over it with you. If you scroll to the last page, all the answers are on the last 3 pages. But I will go through this with you. Just going to wait for about 5 more minutes and let folks come in. It's only 3, 4 of you here right now. Again, if you have anything outstanding, progress reports are available tomorrow. And I'm very happy to say I didn't send out too many letters from this class. So that's a good thing, good thing. Y'all are doing well. That doesn't mean you're not missing anything. That means you're not failing. So good job, good job. But if you are missing something, please make sure that you check on it and get that submitted as soon as possible. But focus on this first. Anything that's due is not due till Friday, if anything. And I think it was only the review sheet that is due this week. I don't think you have anything else due. So unless you have something outstanding, um, this worksheet is not to be turned in. This is just a worksheet. That's why I put it under discussions. So this is not something that'll be turned in for a grade. Okay, I'll give a few more minutes for folks to come in, but in the meantime, like I said, you can look at it. Let me see if I pull it up. Like I said, it's a lot of questions. I don't know how many questions are actually on I know it's more than 50, but again, you're asked to just take your time, read each question carefully. Good afternoon, Andrea. We're doing a worksheet in the discussion, so go, anybody not find it? It's under discussions. It's not under assignments or announcements. It's in discussions. Okay, so we're going to go over these together. Um, and again, I stress that you study vocabulary. You can study this. This is a good worksheet. The worksheet that you had the other day is a good worksheet. Notice this one also starts with organelles. Which of the following organelles contributes to homeostasis by regulating the materials in and out of a cell? And that would be the cell membrane, because remember, I think I put the picture of a cell membrane. Um, Hold on, let me go back for a minute to, let's see, how do we do this? I'm in student view. I think I put it on here. Nope, it wasn't there. On one of the review, let me go back. On one of the review sheets I've included, no, it wasn't that. Or maybe I didn't do it for your class. I need to. All right. Let me include this on this assignment because you really do need to have a visual. Okay. Need to come out of student view to do this. So. Okay, I'm going to add it to. I'm going to add it here because they have a lot of questions about the cell membrane and how it functions. So I want to make sure that I include one that you can glance at. There we go. Okay. All right, let me save this and then I can look at it properly. All right, so I did all of that to say you need to remember to think of the cell membrane as like a screen. Even though it has two layers, it has a bilipid layer, lipid being that it's made of, the molecules are made of fat materials. That it allows, it's semi-permeable because it allows some things to pass through and holds other things outside. But the things that are outside that it wants to take in, these little transport molecules, they are able to bring them in or to send them out as needed. There's two types of transport. We call passing through the membrane transport. So there's two types of transport: the passive transport, which doesn't require energy, and facilitated transport, which does require energy. Okay, so you need to remember that. All right, back to our questions. So I just wanted you to have a copy of a nice colorful copy of the cell membrane. All right, so back to this. So number one is the cell membrane. And remember that word homeostasis that we talked about is going to come up a lot. So make sure that you understand. Here it is again. Which of the following provides the best evidence of reaction of interaction of ribosomes and mitochondria in helping the cell maintain homeostasis? I've seen these questions before. Let's see, let's look over all our answers. All right, energy is required for the cell to produce proteins. So the role of the ribosome is to make the proteins. The role of the mitochondria is to make energy. So that's how those two are related, okay, by their job. Mitochondria is where it produces all the ATPs, which is our energy factory, and the process of making proteins requires energy. So A is the correct answer. All right. And again, these are not going to be the same questions, so it's important that you listen to the reasoning behind why the answer is the answer and not just be able to check off an answer and then go back and try to memorize these questions, because while these are good topics for studying, these are not the questions. Okay, but I'm sure they're similar to what you're going to see. All right, number 3, which of the following describes the interaction between chloroplasts and mitochondria? Again, know what each one's job is. Chloroplast is where photosynthesis takes place, and again, the mitochondria is energy. Chloroplast is D. They manufacture the food. So again, photosynthesis is the process of photosynthesis where plants make their own food. So that's what happens in the chloroplast in a plant cell. We Animal cells don't have a chloroplast. Animal cells don't make their own food. They have to take food in from outside. So that glucose has to travel through the cell membrane to come to the mitochondria in order to get energy. All right, so D is the answer for number 3. And again, if I don't get through all of these, the last page has the answers on it, but I'm trying to go through as many as I can and give you commentary as to why things are the way they are and the way you should be thinking about. It's good. Take your time while you're taking the test. Don't try to rush through it to get it over with. Take your time. Again, this is 20% of your grade, so you don't want to treat it lightly. All right, which of the following evidence of Golgi apparatus and endoplasmic reticulum interacting to allow the cell to maintain homeostasis? Again, the Golgi apparatus is like a storage, it's like a UPS or Amazon, and the endoplasmic reticulum is kind of a highway system. So the correct answer for this is C. When transport vessels are received, chemicals are modified and the resulting products are packaged in the Golgi body for transport to other cells. So it's going to be transported through the endoplasmic reticulum. All right, which two cell organelles work together to help the cell maintain homeostasis by breaking down waste material and producing waste materials produced through cellular activities? And again, the job of the lysosome is to have enzymes that break down things that are ready to be waste products, and then they package it in a vacuole, and then the vacuole moves over to the side of the cell membrane and spits it out. Words that you need to be remember as far as that's concerned: cytosis and exocytosis, things that are coming in and they create a vacuole, into, and things that are already in a vacuole and want to go out are exocytosis, exit, exo. All right, we might run into the word, but I just wanted to put that out there just in case. All right, number 6. The diagram of mitosis is provided. So you have all the stages of mitosis here. And it demonstrates which of the following, and it's A. Mitosis produces cells that are genetically identical to the parents. So you started off with the parent cell that had 2 chromosomes. It went through it made a copy of itself, and the copies separated and created 2 new cells that were also have 2 chromosomes in it, just like the parents. You need to remember PMAT, which is prophase, metaphase, anaphase, and telophase. And those are the phases in order. PMAT. All righty, number 7. Mitosis is critical to cell division and growth because it ensures that, and A is the answer, that every new cell receives an exact copy of the organism's genetic information. That's the job of mitosis. Mitosis happens in all the body cells except the sex cells, the egg and the sperm. And then type in the chat if you need me to slow down, repeat something, re-explain why something is so, because again, you need to understand the process, not because you may not see this question. So if that question made no sense to you and you need me to explain why the answer is so, please do so. Just put a note in the chat. Please stop, go back, say that again. whatever you need to get my attention. I am watching the chat. All right. Mitosis is a critical. Did I do that already? Yes, I did. Number 8. Binary fission is important in reproduction. What form of life? Binary fission, binary means two, and fission means to divide. So it is important for bacteria because this is a form of asexual reproduction. Okay, so if you're able to make notes on your paper to the side, you should do that. Make little side notes. Binary fission is asexual reproduction, and bacteria, single-celled organisms, do asexual reproduction. They just divide in two, and that's how they create new cells. Okay. Guess basically it's mitosis, but then that's their only form of reproduction. All right, meiosis contributes to genetic continuity because it ensures that the gametes of a sexually reproducing organism will contribute half the genetic material of each parent. All right, so that's the difference between meiosis and mitosis, is that you are involving two parent cells instead of one, and you're getting half the information from one cell and the other half to the other cell, so that at the end, the final cell that is made has the full number of chromosomes. Okay? And again, this is for sexually reproducing organisms. Asexually, like we said above, is binary fission. All right. Number 10. The diagram shows meiosis. It clearly represents meiosis because of the following. And C. All right, C is the answer. The cells end up with half the genetic material. All right, so if you see the first one started off with 2 chromosomes, and then they make copies of themselves, and then you end up with 4 individual cells, and you always end up with 4 cells, excuse me, with one, in this case it's 1 or half the number of chromosomes that it started with. Okay. It's always ends because these are the sex cells. So one of these cells is going to combine with one of the 4 cells that as they say this was the female, one of these cells is going to combine with one of the cells from the male, and it will also have 1 chromosome or half the number of chromosomes. So when it unites, it makes a full number of chromosomes. All right, number 11. Which of the following are large molecules that are broken down into smaller molecules during digestion? And it is carbohydrates. And all the rest of these are the smaller products of macromolecules. So when you break down a carbohydrate, you get simple sugars. When you break down lipids, you get fatty acids and glycerol. When you break down a protein, you break it down into amino acids. So these are all the units that go into making their macromolecule. Okay. And again, you have a chart, and that was one of the questions on your chart was, what are the building blocks of that macromolecule? So carbohydrates are made from simple sugars. 12. The surfaces of the leaves of many plants help to reduce water loss because they are not water permeable, meaning the water doesn't go through. How does the biomolecule that makes up the surface differ from other biomolecules? All right, it is made up of long chains of fatty acids. You know, water and fat doesn't mix. Okay, so the layer you ever touch the top of a plant, it's like a waxy surface. It's made up of long chains of fatty acids. and that makes it kind of waterproof. Okay, they get the water through their vascular system. So the water doesn't land on top of the leaves and get soaked in. It comes up through the vascular system that it has internally. That's how they get its water. Okay, if you ever tried to water your plants and it gets on top, it kind of just rolls off. And so that's why. Alrighty, 13. Which of the following are molecules that make up muscles and organs and transmit signals between cells and constitutes immune molecule? So that means this molecule does a lot of things. So that's proteins. Almost all of your body is protein. What part of it isn't a protein is water. Okay, so the proteins make up hormones. The proteins are found on your muscle cells, your organ cells. They're all made of protein, which is why ribosomes are real important, which is why when you look at a cell, there's a lot of these little dots all over the cell because you need to have a lot of factories making proteins because everything is made of proteins. All right, which of the following biomolecules is a major energy source? That's uh, okay. That one could be your worksheet is going to say that it's lipids, and lipids are they do have more energy than a carbohydrate. It's just harder to get to a lipid. It's easier to digest carbohydrates, so you get quick energy from a carbohydrate, but there is more energy in a fat molecule than there is in a carbohydrate. Okay. So I just said that for both of these. It depends on how they ask the question. But like I said, your answer sheet says B. And to be honest, all of these except nucleic acids, proteins also provide energy. If you're on a diet, the whole reason the diet works is because if you don't give it carbohydrates to get energy, it's going to take it from the fats. And if you run out of fat molecules for it to take energy from, it'll take it from the protein. So it's not, it's the proteins are because it's tissue, is its last resort. The body's smart like that. But it can get energy from that. So this is why people kind of waste away when they don't eat, is because you can get, the body will find a way to get energy where it needs to get it from. So it will get it from the proteins if necessary, but you get a whole lot more energy in a fat molecule than you will a carbohydrate. All right, 15, which is a function of a lipid, and they are to store energy. In which one? Okay, D. Store energy for the cell. So that goes back to 14. Which, all right, 16. Which of the following is a function of enzymes? Enzymes increase the rate of reaction. They don't become a part of the product. So they are necessary. Some reactions don't take place unless the little instigator molecule gets involved, and that's what enzymes are. They make reactions happen, but they don't become part of the product. Once they get the reaction going, they leave. All right. When you do chemistry next year, you'll talk more about how that works. All right. The difference between active and passive transport, excuse me, is active transport requires energy, mentioned this earlier, to move molecules from a concentration gradient, and passive transport does not. Concentration gradient is like your cell membrane, and they had the pictures of the beaker with the little line in the middle. And remember, the concentration goes from an area of high concentration to an area of low concentration, always. And that's diffusion of molecules. And then if it's diffusion of water, it's called osmosis. And I get ahead of myself because they're going to ask you that right now. All right, so you have a picture here. You have a picture, so study the pictures, study all pictures that they give you before you try to answer the question. Before you even look at the question, study the picture. So you've got little molecules, little dots on the outside, and then you've got a cell membrane, and then you've got these big transport molecules that are sitting in the cell membrane. And then you got some dots on the inside, which are some more solute. So automatically, before, like I said, before you even read the question, what you should deduce from this is that there are molecules outside the cell and they want to get inside the cell. There are a lot more molecules inside than outside, which means the concentration is high inside and low outside. So the molecules are going to want to move from high to low. They have molecules, which tells you that they are past, the way that they're getting into the cell is through these transport molecules. So look at the arrows. There are arrows on this diagram. They're getting in through the transport molecules. All right, an illustration of a cell interacting with its environment is provided. The illustration provides the best represents which of the following. All right, and it is B, active transport of solute into the cell using energy. And we say active because they have to, the only way those things are going to get inside is through that carrier protein. And the cell has to actively allow that to happen. Okay, the carrier proteins have to open and close to bring things in and out. That requires energy. That's active transport. All right, 19. Which of the following most accurately describes the difference between osmosis and diffusion in the cell? Okay, both are diffusion, but osmosis is A, the movement of water across a membrane. Diffusion is the movement of molecules from high to low. Okay, so something is moving across the cell membrane, and it's moving from high to low always. You might hear things like reverse osmosis. When they want to force osmosis to go in the opposite direction, meaning go from low to high, then they produce a situation which is called reverse osmosis. All right, 20. Active transport maintains homeostasis in the cell through which of the following processes? By using energy to move molecules from where there are more of them to where there are fewer of them. That's another way of saying from high to low. Okay? So again, active transport from where there's a lot of molecules to where there's a little bit of molecules. And I would say this picture up here too. The reason why you have to actively move those things in, because the tendency would be to try to push, because these arrows are pointing into the cell. This is active transport because the tendency is to bring something against the concentration. You want those molecules inside, even though there's fewer of them outside than inside, it's not going to flow naturally. They have to force them to come inside. It's active transport. All right. Here again, you want to look at the picture before you do anything. So you have a barrier in the middle of this U-tube. The barrier represents like a cell membrane, and it tells you a high concentration of sugar, a low concentration of sugar on the left side. So those are those little dots. There's fewer of them on the left than there are on the right. Okay, and you got a membrane in the middle. Water with a concentration of sugar is placed in one of the containers. The water with the higher concentration of sugar is placed on the other side of the container. The two sides are separated by a barrier. This situation will demonstrate passive transport if which of the following occurs. So again, passive means it'll pass with requiring no energy at all. And so they will pass if the sugar molecules can pass through the barrier until the concentration of sugar is uniform throughout. That's A. Okay, so if those holes are small enough, all those molecules will try to go to the other side. And diffusion doesn't happen forever. It'll stop moving when there's an equilibrium. So when there's an equal number of molecules on both sides, diffusion stops. And we call that equilibrium. Alrighty. Again, stop me if I'm saying something that sounds totally foreign to you. Because these are made up from a Georgia milestone test. So please remember that part. This might not be the test, but these are milestone questions. All right, which of the following provides the best description of the role of glycolysis in cellular respiration? And again, we did some of these so far back. Yes, it is. Absolutely. So rather than you go through Canvas and pull all those other study guides together, this one piece of paper is probably the best thing that you could do. All righty. Took a minute for me to find one, but I found it. All right, back to glycolysis. So respiration has different stages to it, and glycolysis is one of the stages. And B is the answer to that. It breaks down the glucose and releases the pyruvic acid. All right, again, if you go way back and you have this in, if you look this up in Canvas, glycolysis, you have pictures. Glucose is a 6-carbon sugar. And what it does is it breaks it down into 2 3-carbon molecules. Okay, so that's what this process is doing. And that process is called glycolysis. And it's on the way to making the ATPs. All right, what is the relationship between photosynthesis and cellular respiration? And this is the one I pointed out last class with the equations. The reactants of one are the products of the other. So remember that one. If you're looking at photosynthesis, it's carbon dioxide plus water yields glucose and oxygen. If you take that same equation backwards, it's oxygen plus glucose gives you carbon dioxide and water. Okay, so that's what they mean by the reactants of one. So photosynthesis would be water and carbon dioxide, and the products of that are glucose and oxygen. The products of that now become the reactants for respiration. So your glucose and oxygen are your reactants, and the products are now carbon dioxide and water. All right, so again, just kind of have a visual on that equation and know it forwards and backwards. All right, again, this is a picture, it's kind of like the one you had in your worksheet. Provides an incomplete concept of cellular respiration. What are the missing components that are labeled? So what goes in, so like what I just said, what goes in are the arrow, what are the reactants? So oxygen is one, what's the other word? You tell me, which is the right. What's the other word? Something plus oxygen goes in during cellular respiration, and we get heat in the form of ATP, water, and something else coming out. I just told you this. What's the answer? Which one is the answer? All right, yes, B. Thank you. So glucose plus oxygen goes in, and carbon dioxide and water come out plus ATP. All righty. And again, if you pull this up. You can write notes on, do it and help you download it in such a way that you can scribble on it and add commentary and other notes to it. Okay. You've got a picture, and I need to make mine a little bigger because that's too small for me. Okay, hold on. Okay. All right, so you've got time. Let me make it bigger some more if I can. That's as big as it's going to go. All right, you've got 8 hours of sunlight in the first one, 6 hours, 4 hours, and 2 hours. So you're having an experiment where they're being exposed to sunlight. You've got plants, two plants in the first one, one plant in the second one, two plants, and one plant. So the student performs an investigation on the 4 beakers of water that are set up under different conditions of plants and sunlight. The water in all 4 beakers began with the same amount of dissolved carbon dioxide. The most reasonable prediction at the end of the investigation is the beaker with the lowest amount of carbon dioxide will what? I mean, which beaker will have the lowest amount of carbon dioxide? So that goes back to our equation for photosynthesis. What do you need for photosynthesis? It's actually 2 things you need for photosynthesis. So you kind of have to, and I'm going to tell you it's going to be beaker A. One, because you've got 2 plants in there, and so that's twice. You need carbon dioxide for photosynthesis. And there's 2 plants in there using up all the dissolved carbon dioxide. It's A and not C, even though you have 2, because you need sunlight. Photosynthesis does not happen without sunlight. You have more sunlight hours on A than you have on C. C only has 4. So it's not that it's not going to happen, and it's going to happen in all of them, but there'll be less carbon dioxide at the end of the time in beaker A because there's 2 plants and you've got more sunlight, so the reaction is going to happen faster. Okay. Again, the sunlight energy acts like a catalyst. It makes the reaction go. The reaction is going to go a whole lot better if you have more light. Okay. Again, so the note with this one, the way to answer this question is to think about photosynthesis. You need carbon dioxide for photosynthesis. And that's why they pump the carbon dioxide in the beaker so that those leaves can use it to photosynthesize. All right, 26. Which statement correctly describes how the structure of DNA facilitates replication? All right, the answer is B. When you have a DNA molecule, remember that it's going to unzip, and only a portion of the DNA is going to actually replicate. The whole thing does not replicate. So if you go back to the pictures we had in your notes, it unzips, and a portion of the unzipped DNA is replicated. So that's what they mean by only a few nucleotides, and there's only a limited variation is possible. All right, 27. I hope you don't get that question. Which of the following provides the best evidence to support the claim that non-disjunction has occurred during the process of cellular division? All right, non-disjunction is one of those vocabulary words you're going to have to make sure you look that up, but it has to do with the chromosomes being able to, what it's supposed to do is when the pairs are supposed to separate, and when they don't separate, It says one C is the correct answer. One daughter cell has both of the chromosomes, the other has neither. So when the cell separates, one pair is supposed to go one way and the other is supposed to go the other way. And sometimes they don't separate. So both of them are in one cell and there's nothing in the other one. So that's non-disjunction. All right. We'll take a break. Anybody need to go to the bathroom or something? Ah. Which of the following is the best explanation for the process of translation? And I believe we were talking about that the other day. The basic structure of DNA is changed due to a mistake or error in environmental factor. All right, the first one's not going to happen. All right, here again, they picked D as the answer. I'm not sure. Translation is when I don't know why they picked that as an answer. I would have picked B. Because remember the other day we talked about translation and transcription. You have to transcribe it, make a copy of it from the messenger RNA copies the information, so that copy is transcribing, and then it takes the information to the ribosome where it is translated. The only way you're going to have a mistake is if it transcribes it wrong, okay? Or if there was a defect. It can skip a nucleotide in which it would have transcribed it wrong. Actually, it'll transcribe it correctly, but it did it in such a way that it skipped one, or one is missing, or one is added. It puts it in a sequence that is not normal, and then so it'll give you a different code. So I'm not really liking that question or the way they answered it, but so put a star next to that one. Like I said, the answer key says it's D, but I think I, D, it's not the way they asked the question. It should be which of the following best explains why there would be a change in the process of translation, and that way D would make sense to me. All righty. This is an ecology question, so this would have been one of those questions that we didn't quite get to. So they have population X is exposed to radiation, and you get a mutation. Okay, this could be a DNA question. Population Y is shielded from radiation, so there's no mutation. So the offspring um, end up in the generation 31 look like the original. But in population X, the offspring are going to be changed in a slight way. The picture doesn't show it real good, but they're implying that there's a change. In the investigation, involves species of microorganisms. One population is exposed to radiation, the other population is shielded. The population exposed to the radiation exhibits a mutation, while the other does not. Both populations are allowed to reproduce under normal conditions. 30 generations later, the descendants of population exposed to the radiation still exhibit the mutation. Why can't this observation be used to support the claim that exposure to radiation did not cause the mutation? Why would you think? And the correct answer is actually D. In the first generation, or we call it the second generation, actually, when you have the mutation, while there's no more exposure after that, that mutation is all that there is to be passed on to the next generation. So it's going to keep passing on that mutation every time, even though it's not exposed to any more radiation. That cell is what it is, and that cell will continue to reproduce whatever the mutation was. So once the mutation is present, it is inherited by all the offspring in every generation. Because remember, microorganisms do binary fission. They don't combine with other things. They just divide to make a copy of themselves, and they divide in two. So they will keep repeating that defect over and over again. All right, changes in DNA such as insertions or deletions. Insertions means you added a nucleotide, or deletions of nucleotide was left off, are likely to give appearance of new traits. Because remember, if you leave a nucleotide out, then it's like spelling your name wrong. Okay, you might be able to read the name, but it's spelled wrong, so it's going to have different characteristics. Okay, so remember the protein, if it leaves one out, it's like adding a letter to your name or deleting a letter from your name. It's going to be different. All right, 31. Genetic engineering was a major factor in the development of which of the following? And somebody did their project on this. Disease-resistant crops. Okay, or GMOs. So the whole point of genetically engineering crops is so that they don't catch disease, that it's not tasty to the bugs, and that's why they do it. Supposedly so that we have more crops to feed more people. Alrighty. Tomorrow's Earth Day. And if y'all weren't testing, I'll do this with you all next week because I don't want to take up your review time. But one of the topics that we talked about is feeding more people, sustainability. So I'll talk that with you next week. But that is the major focus of Earth Day this year. Excuse me, is a sustainable future by creating more food and better crops and just stuff like that. All right, 232, one product of biotechnology is genetically modified food, GMOs. Some people are concerned about these because the production of these food increases the biodiversity of organisms on the earth. Um, Again, that's the answer that they gave you on the worksheet. But it's better than all the other choices. One of the concerns that people have about genetically modified foods is the body recognizes it as different. A lot of times, for example, with salmon that they genetically modify, they become sterile, they don't reproduce. Your body will recognize it as a foreign substance and it may not digest it properly, it'll store it as fat. So those are the real concerns with genetically modified foods. Again, they do it so that they can produce production these things. I don't know about biodiversity, but they do it so that they can increase the production of food, and that's why they do it. All right, which of the following is one of the ethical concerns raised by cloning? And again, cloning means that you created an exact copy of a living thing. And it's a potential life is destroyed when the potential life is destroyed when the embryonic cells are used. I don't know about that, but that's the answer they give you. Technology is required. Uh, I don't know. You would have to know more about the process as far as embryonic cells being a life being destroyed. All they're doing is removing the embryo from an organism and placing it with another. So I don't know about it being destroyed. They just another question like that I could question. So hopefully you won't get one like that. All right, which 34. One of the ethical concerns about stem cells is that C, some are created by destroying human embryos. Yes, that's a better answer than this one up here. Um, they get them from certain embryos, and then the ones that they remove it from have to be destroyed and discarded. So that makes sense on that one. Not so fond of the one before, but this one does make sense. All right. As advances are made in biotechnology, it is important to make informed decisions to avoid hazards associated with which of the following? And it would be A, dangerous organisms. You don't want to create a monster when you're tinkering with cells. That put me in mind of not the TV show the other day, 9/11, where she was doing a vaccine and she decided to alter the vaccine so that it worked faster, and all it did was create a pandemic. So you can create by trying to do biotech can be dangerous if not put in check. All right, which statement describes the role of meiosis during reproduction? And that is C, during meiosis, pairs of chromosomes exchange genes producing genetic variation. So again, this is why, because of meiosis, we don't look exactly, we're not clones. We don't look exactly like our parents because there is genetic variation. But not during mitosis. Mitosis, if you're making a skin cell, you're going to make a skin cell that'll look exactly like the other skin cells. Imagine if you had variation when your skin cells and organ cells divided. You'd be looking different every other day. And also remember, they haven't asked it yet, but that they should. Cancer. Cancer is a result of cells dividing uncontrollably, and then they grow in places where they don't belong. Okay, instead of it just having normal cell division, they continue to divide and divide and divide until they create growths. And usually that growth has some type of biological interference. So that's what cancer is. It's just abnormal cell growth of tissue in places where you don't want that growth to happen. All right, Mendel's law of independent assortment is best demonstrated with which of the following observations? And it would be when two sexually reproducing organisms produce offspring and each offspring has unique set of genes. Again, there's that word independent assortment, and that was kind of like the other word we had, non-disjunction. You want to create variation so that the offspring don't look exactly like the parents. Okay, so the gene, this chromosome might have went to this cell and that chromosome went to this cell. They both didn't go to the same cell. That's what that's saying. All right, during meiosis, genetic recombination results from chromosomes assorting independently or crossing over. Crossing over is where a little tip of the chromosome kind of exchanges places with the other one so that when it's copied, a different chromosome is copied when they go through the process of copying, replication. All right, Mendel's law of segregation. So yeah, Mendel has a couple of laws that you have to remember: independent assortment, segregation, and I think there's two other ones. Is absorbed during the stage of mitosis, which the sister chromatids are pulled apart and two cells are divided into four. Okay, so segregation, pulled apart, kind of associate those two words to help you answer that kind of a question. And again, testing skills, just associate words in the question with whatever the word is in the answer. So if you're guessing, that's called an educated guess, where you might not have a clue, but try to make the words in the answer match the words in the question. Segregation and pulled apart, they go together. All right, which of the following characteristics of meiosis explains the process that contributes to genetic diversity? And diversity means differences. That would be A. Meiosis results in novel combinations of alleles of and I don't like the way they answer that. But anyway, this is when they say novel, that's like saying unique. Okay. Results in now different combinations of alleles to create offspring. You don't look exactly like your sister unless you're twins, sister or brother, because there are unique combinations that came from those same two parents. All right, Punnett squares, Punnett squares. And I think we can get two more questions done before we have to go. All right, so I'm going to do this one and the next one, and then we'll stop. All right, so we're looking at the square. Hopefully everybody remembers how to do a Punnett square, where the letters at the top represent one parent, the letters on the side represent the other parent, and then you're going to take one allele. These represent alleles of a particular trait. One from the mom, one from the dad goes in each box. This is a probability, and each box is 25%, and the whole box makes up 100%. Okay. All right. The gene for tallness, and they use the letter, other rules that you got to remember. They use the letter of the dominant characteristic is always capital, and they use that same letter to represent the recessive character. Even though short starts with an S, we're going to use T because T is for tall. So capital T for tall, small t for short. Okay. And usually the question will tell you which one is which. The predominance, the Punnett square shows, and it says below, but it's above, shows a cross between two tall pea plants. All right, this has a phenotype ratio, and these are they're tall, but they're heterozygous tall. Okay, so they're carrying a gene for shortness, although the plant itself, when you look at it, is tall. Okay, the phenotype ratio of the offspring produced. So we're reading the box. So the first box is homozygous tall, and there's only one of those, so that's 25%. There's two of the big T, little t, so that's 50%, and there's only one of the little t, that's 25%. But the heterozygous ones are tall, so basically we have 3 tall to 1 short. So it's going to be A, 3 tall to 1 short, even though they're heterozygous and one's homozygous. They're tall because they ask you about phenotype. What are they going to look like? If they ask you genotype, that's different. Phenotype means what do they look like? 3 out of 4 of them are tall. All right, number 4. In fruit flies, the gene for eye color is located on the X chromosome. So that is a sexually characteristic, sex-linked characteristic. And the gene for red eye R is dominant over the white for eye color. So again, they use a capital R and a small r. Red is dominant, white is recessive. The female fly, and that the X is for female and the R is for the eye color. And then has another trait. So this female, because you got two X's, but they are homozygous for eye color. It's mated with a fly, a male fly. So that's why you have X and a Y. And because the gene is only located on the X chromosome, you don't have another letter on the Y. Okay, so that's why you only see one R. All right, which statement best describes the expected outcome of this cross? All right, so if you did a box like that, like the one above, you would find that the chances of getting a male, it's going to be similar to the one above. The chances of getting a male that is that has the answer is B. You're going to get 25% of them are going to look like this. They're going to be male with a dominant R. And you're just going to have to draw that out so that you, and I'll draw it out for next class because we are running out of time. But I would draw this one out. And this is one of those boxes where you can associate the R and the big R and the little r with your X and Y. And then you'll know which ones will go in which box. So I'll draw that one out for you for next class. We might be able to get one more done. All right, in certain rose plants, white roses and red roses are incompletely dominant, which means they share it. It's neither white nor red. When the red rose is crossed with a white rose, a pink rose will result. What is the probability of producing more pink roses than white roses? 50-50. And again, I'm going to draw these out for you for next class. All right, I'm going to stop right there because that's a lot. So we got through half of it, just about. And I will do a visual for that, but you can try practicing that one yourself. And I'll have that for you next class. But practice your Punnett squares. Some of you weren't real good with doing Punnett squares, so make sure that, and you'll get scratch paper for the exam so that you'll be able to do questions like this. You can do on the side so that you can see the results of the cross and then better make a educated guess as to what the answer should be, because these you actually do have to do. You can't just guess at them. You actually have to do a cross in order to figure out what the answer will be. Okay, so you will get scratch paper and things. You can't take notes into the exam, so remember that. So you please study. And I'm going to stop right there. All right, any questions? Any questions? Any questions? All righty. All right, that being said, like I said, I'm going to stop, and you all have a great rest of your day. Enjoy the sunshine. For now, it's supposed to rain, storm tomorrow. But enjoy the sunshine today. All right, bye-bye. Okay.